Okay, so welcome to this next video in which we are discussing the VIP and PACAP receptors. Okay, right. So what we have just discussed is the fact that all of these three receptors that are in this family of VIP and PACAP receptors are G-protein coupled receptors. Specifically, they're secretin family G-protein coupled receptors, which means that they bind their ligand in that slot between the amino terminal domain, which is piled on top, and the transmembrane domain, which is below. Okay, right. Now, we want to now see, once the receptor has been activated by ligand binding, what's it going to do? Okay, what's it going to interact with? And to understand this, we need to discuss the heterotrimeric G proteins. Okay, right. So, heterotrimeric G proteins, what does that mean? Well, trimeric means that they're going to be made out of 3 subunits. Heterotrimeric means that they're going to be made out of 3 different subunits. So these are G proteins that are made out of 3 different subunits. Okay, so we're going to start off by discussing the most important subunit of heterotrimeric G proteins, which is the alpha subunit. Okay, so I'll draw the alpha subunit here, and we'll colour it in, I think, in red. Okay, so here in red, this is the alpha subunit of our heterotrimeric G protein. Now, alpha subunits are special because they can be in 2 states. They can be in an on state, in which they have guanosine triphosphate GTP bound to them, and they can also be in an off state in which they have guanosine diphosphate GDP bound to them. Okay, so we'll start off with our alpha subunit being in the off state with guanosine diphosphate bound to it. Okay, now the other thing that's important to discuss is that the alpha subunit is not an integral membrane protein. It's not sitting within the membrane. Okay, so here is our phospholipid bilayer, by the way. That's what these 2 lines are. This is the inner leaflet, this is the outer leaflet. However, it is attached to the inner leaflet of the phospholipid bilayer. So how does it achieve this? Well, basically, it has a lipid moiety sticking off it, and this lipid moiety sticks into the inner leaflet of the phospholipid bilayer and holds the alpha subunit at the inner leaflet of the phospholipid bilayer. Okay, so the usual lipid moiety that you have here is what's known as a palmitoyl group. Okay, so I want to discuss this in a bit more detail. So we're going to discuss the process of S-palmitoylation, which is the main way in which lipid moieties are added onto alpha subunits of heterotrimeric G proteins. Okay, right. So we'll start with what palmitic acid is then. Okay, so palmitic acid is the old name for a molecule that would now be called hexadecanoic acid. Okay, and although hexadecanoic acid is a bit more of a mouthful than palmitic acid, it's a useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 16-carbon, fully saturated carboxylic acid. Okay, so let's show this here. Here's the carboxylic acid group, and then you're going to have 14 methylene groups in the middle. Now, because I don't want to have to draw 14 methylene groups, what I will do is draw 1 methylene group. I'll then bracket it like so, and I'll subscript this bracket 14. Now, this is a useful trick. What this means is repeat this structure 14 times, okay? So it gets you out of having to do a lot of tedious drawing. Then we have a methyl group right on the end here. Okay, so that takes us up to 16 carbons in total. We've got the 14 in the middle and the 2 on the side. That's 16 in total. Okay, right. So this is a very long molecule, and most of it is extremely hydrophobic. So it's not going to interact well with water, but it will interact beautifully with the hydrophobic core of the phospholipid bilayer. So once you stick this molecule onto the side of your alpha subunit, you'll have this tail dangling off because it's the carboxylic acid group you're going to use to attach it on, okay? And this will then interact very poorly with the water in the cytoplasm, so it results in this lipid moiety ending up sticking in the phospholipid bilayer and holding the alpha subunit of the heterotrimeric G protein under the inner leaflet of the phospholipid bilayer. Okay, so how do we attach these palmitic acid molecules onto the alpha subunits of our heterotrimeric G proteins? Well, basically, the way that you always add palmitic acid molecules, well, actually it's not true. There are two ways that you can add palmitic acid molecules onto proteins, okay? One is S-palmitoylation, the other is N-palmitoylation. N-palmitoylation is very rare, okay? S-palmitoylation is the main one. Okay, and it's certainly the one that's used in alpha subunits of heterotrimeric G proteins. So, basically what you do is you stick the palmitic acid molecule onto a thiol group that is within the R group of a cysteine residue. So you attach it basically to a cysteine residue within the polypeptide of the alpha subunit. Okay, so let me show you this here. So, let me firstly draw the core amino acid structure. So here is our core amino acid structure. We've got the amino terminal over here, we've got the alpha carbon here, we've got the carboxylic acid terminal here. Okay, and then of course I've drawn it as though it's involved in a polypeptide, so I've drawn it as though we've got an amino acid in front here and an amino acid behind here. Now the R group of a cysteine amino acid consists of a methylene group, like so, with a thiol group then coming off. Okay, so when you have a sulfur atom with a hydrogen atom coming off like that, that's called a thiol group. And it's similar in its nature to an alcohol group because sulfur atoms have similar chemical properties to oxygen atoms. Okay, sulfur is in the same group of the periodic table as oxygen, it's just underneath it. Okay, so if that was an oxygen, of course, this would be an alcohol group, and since the sulfur has similar chemical properties to the oxygen, we can therefore expect that a thiol group like so will have similar chemical properties to an alcohol group. Okay, so basically you can take a long-chain carboxylic acid, such as palmitic acid, and you can react it with a thiol group, just like you would react a long-chain carboxylic acid with an alcohol group. Okay, so what's going to happen is you're going to take the alcohol group off the carboxylic acid group of the palmitic acid molecule. You're going to take the hydrogen off the thiol group. You're going to bind those two things together to make water. Okay, and you're then going to bind this carbon of the carboxylic acid to this sulfur of the thiol group, and that link that you then get is analogous to an ester link, but is of course involving a thiol group, not an alcohol group, so it's called a thioester link. Okay, so you attach palmitic acid molecules onto thiol groups via thioester links. Now, we have not added the full carboxylic acid molecule onto the cysteine residue of the alpha subunit of the heterotrimeric G protein. Instead, we've added everything but the alcohol group. We've added this portion that I am now circling in green. Okay, now the name for this portion that I am circling in green is a palmitoyl group. Okay, so it's not a molecule. This is not a molecule because it's not finished. This carbon doesn't have a bond. Okay, it needs a nut to be added onto something to finish it. Okay, so it's called a group rather than a molecule. It's something that you can stick onto the side of something else, and indeed we're going to stick it onto the side of this cysteine residue. So this is called a palmitoyl group, and the process of sticking a palmitoyl group onto something is called palmitoylation. Since we're going to stick the palmitoyl group onto the sulfur of a thiol group or of a cysteine residue, this is S-palmitoylation. We are palmitoylating that sulfur atom. Okay, right. So every single alpha subunit of the heterotrimeric G protein has a palmitoyl group attached onto one of its cysteines. So there is a little bit of evidence now that some of them might have two palmitoyl groups. Okay, so they'll have two cysteines which have got palmitoyl groups attached to them. But every single alpha subunit has at least one palmitoyl group added on. Most of them just have one. Okay, right. Now, some alpha subunits also get another lipid modification occurring to them. Some of them also undergo a process known as N-myristoylation. Okay, so let's now talk about what N-myristoylation means. Okay, so it's a very similar process. This time you add on myristic acid rather than palmitic acid. Okay, so what's the new name for myristic acid? Because myristic acid doesn't tell us anything. The new name for myristic acid is tetradecanoic acid, and then everything is suddenly simple. Okay, so this is a 14-carbon, fully saturated carboxylic acid. Here's the carboxylic acid group, then we're going to have 12 methylene groups in the middle. Okay, so here is 1, and again I'm going to use my trick of bracketing it, then putting a subscript 12 there, and then a methyl group on the end over here. Okay, that's the structure of a myristic acid molecule. Okay, and this time we are not going to add them onto cysteine residues. Instead, you add it onto the amino terminus of your polypeptide. Okay, so let me explain this. Here, let's say, is the polypeptide of our alpha subunit. It's a polymer of amino acids, and it has an amino terminus, which has a free amino group, and a carboxylic acid terminus, which has a free carboxylic acid group. Okay, now, you are going to bind this carboxylic acid group of the myristic acid molecule to the free amino terminus of the first amino acid along. However, you can only do this if the first amino acid in your polypeptide is a glycine amino acid. Okay, and the single letter amino acid code for glycine is G. Okay, so you might be sitting there thinking, wait a second, this is rubbish. I thought the first amino acid of proteins was always methionine. And I would say you're completely right. When you first translate a protein, okay, so when a protein first emerges from a ribosome, the first amino acid in that polypeptide will absolutely always be methionine. Because that's the amino acid that corresponds to the start codon, okay? So mRNA, you begin translation of mRNA with a start codon, AUG, okay? And the corresponding amino acid that is put in is then a methionine, so you can't get around the first amino acid being a methionine. It's called the initiator amino acid. Okay, however, after translation is completed, often that methionine's clipped off, basically. So that's how you can end up with the first amino acid being a glycine. Okay, so if your first amino acid is indeed a glycine, then you can have myristic acid molecules attached onto the free amino terminus of the polypeptide. So let's draw this. So here then is the amino terminus that is free. Here is the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group. Now, what's the R group then of glycine amino acid? Well, it's the simplest proteinaceous amino acid, just meaning the amino acids used in proteins. And it just has a hydrogen off the side. Okay, so this is our glycine at position 1. And basically what you can do is you can form an amide link between your myristic acid carboxylic acid group and your amino group. So what's going to happen then? Actually, I think I'll draw the myristic acid molecule again down here, rather than somehow trying to cross across the peptide that I've drawn here. Okay, so here is our myristic acid molecule drawn again. It's a 14-carbon, fully saturated carboxylic acid. There are 12 methylene groups. Here's our methyl group right on the end. Okay, and what's going to happen is you're going to form an amide link between the carboxylic acid group and the amino group. And all you do is a condensation reaction. You take the alcohol group of the carboxylic acid group, you take the hydrogen, well, a hydrogen of this amino group, combine those two together to make water, and then attach this carboxylic acid carbon onto that nitrogen atom there, and this link here is called an amide link. Okay, right, so that's how we're going to attach on our myristic acid molecule onto the amino terminus. Now, it's the same story. We haven't actually attached on the full myristic acid molecule. Instead, we have attached on the myristic acid molecule minus the alcohol group of the carboxylic acid group. Now, that group now, because it's not a molecule anymore, it's not finished, you need something attached onto this carboxylic acid carbon. This group is called a myristoyl group. Okay, and the process of adding myristoyl groups onto something is called myristoylation. Okay, and since we are myristoylating an amino group, we are myristoylating a nitrogen atom, and therefore this is N-myristoylation. Now, some alpha subunits of heterotrimeric G proteins get N-myristoylated as well as S-palmitoylated, okay, which means that they have two lipid moieties sticking off the side. They have a myristoyl group sticking off their amino terminus, and they also have a palmitoyl group sticking off some cysteine somewhere. Okay, right. Now, which alpha subunits then do indeed get N-myristoylated? Well, basically it is the alpha subunits which are within the G alpha I/0 family of alpha subunits of heterotrimeric G proteins. Now, we will come to this. We are very soon going to discuss all the different types of alpha subunits of heterotrimeric G proteins, and we'll discuss the G alpha I/0 family, which is one of the four families we divide the alpha subunits into. There are actually 8 different alpha subunits, well, in fact, 8 different genes for alpha subunits that they code for more than 8 different subunits. But there are 8 different genes within here, and all of the alpha subunits they produce will end up being N-myristoylated for the simple reason that they have a glycine in the correct position. Okay, whereas the other alpha subunits can't be N-myristoylated because they don't have the correct glycine at position 1. Okay, right. So those family of alpha subunits, they get 2 lipid modifications attached to them, basically. So they get a double whammy of this, and they most definitely implant into the inner leaflet of the phospholipid bilayer, and hence the alpha subunit is attached nicely underneath the cell membrane. Okay, moving on. The next 2 subunits of the heterotrimeric G protein always stay together, okay, so they never ever come apart under physiological circumstances, and they're often collectively known as the beta-gamma complex. Okay, because they never ever come apart, and they are of course called the beta subunit and the gamma subunit, and because they always remain together, the complex of the 2 of them is called the beta-gamma complex. Now, I should say, when the alpha subunit is in the off state, okay, when it has GDP bound, it combined with a beta-gamma complex. However, when it's in the on state with GTP bound, it doesn't associate with a beta-gamma complex, and that will become important later. Okay, so here is our beta-gamma complex here. This is a beta subunit, this is a gamma subunit. So we'll colour in gamma in green, G for green, okay, and we'll have beta in blue. Okay, right. Now, the beta-gamma complex also has a lipid moiety attached to it. Specifically, the gamma subunit of the beta-gamma complex has a lipid moiety attached to it, which sticks into the inner leaflet of the phospholipid bilayer, and hence holds the gamma subunit to the inner leaflet of the phospholipid bilayer. The beta subunit does not have its own lipid moiety. It relies on being attached to the gamma subunit, which is in turn attached to the inner leaflet of the phospholipid bilayer. If they did come apart, beta would no longer stick to the inner leaflet of the phospholipid bilayer. It would fall into the cytoplasm, basically. Okay, so it's a good job they never do come apart then. Okay, so this lipid moiety here, it's the same principle as the palmitoyl and myristoyl groups that can be added onto alpha subunits, but it is a different type of lipid moiety. Okay, it's a geranyl group, which is also added onto cysteine residues. Okay, so on a cysteine residue within the gamma subunit, you have added something called a geranyl group. And I think I might try and squash in the structure of one of these down here. Okay, so let's draw a cysteine then. Here's our amino terminus, here's our alpha carbon, here's our carboxylic acid group, and again I'll draw it in residue form, so I've drawn it as though it's attached to neighbouring amino acids. Then we've got a methylene group with a thiol group coming off it. Okay, and then attached to this sulfur atom you're going to have a geranyl group. Now, I do not have room to draw this in molecular format, so I'm going to draw it in skeletal format. So remember, in skeletal format you don't show carbon atoms, they're implicitly shown by corners, and you also don't show hydrogen atoms coming off carbon atoms. When there are missing bonds coming off carbon atoms, it's implicitly understood that those are two hydrogens. Okay, right, so this is the structure then of a geranyl group. So that's a corner, that's a carbon. Okay, here's another carbon, another carbon. We're going to have a double bond there, like so. And then we're going to repeat this structure. Okay, so now we repeat it exactly the same. Okay, so I'll try and highlight this up for you. So, this is one here in green. Okay, there. Now we're repeating that same structure here in orange. Oh, it's smudging nastily, but never mind. Okay, so we've got a repeat of that. We're going to repeat it again, and we're going to repeat it a second, we're going to repeat it a fourth time after that. Okay, so there's a repeat of it, and then here is our next repeat of it. Okay, and I'll get some colour on there as well. So here is another repeat of this structure. Okay, and then finally we have another one. Okay, so 4 of them overall. That makes our geranyl group here. Okay, right, so this is a very hydrophobic structure again, and this is going to nicely implant into the inner leaflet of the phospholipid bilayer and hold our gamma subunit to the phospholipid bilayer, and since the gamma has the beta attached to it, it holds the whole beta-gamma complex at the inner leaflet of the phospholipid bilayer. Okay, so when the alpha subunit is in the off state, you therefore have the 3 subunits all associated together, and here we have the heterotrimeric G protein. Okay, so we'll have a break there, and in the next video what we'll do is we'll begin by discussing all the different types of alpha subunits, all the different types of beta subunits, and all the different types of gamma subunits, so that we have a complete understanding of that, and then we'll move on to the G protein cycle, and then we'll discuss more specifically the heterotrimeric G proteins, which are going to be activated by our 3 VIP and PACAP receptors, and the downstream signaling pathways which follow.